Welcome to the presentation. The research paper title is Identification of Human HK Genes and Gene Expression Regulation Study in Cancer from Transcriptomics Data Analysis. The research paper aimed to understand which cell type-specific patterns of gene expression differ among different cell types, and particularly for cancer. Also to define the gene expression pattern from 3 directions and categorize cancer HK genes to observe gene expression regulation in cancer cells. A housekeeping HK gene is an essential gene that is required for the maintenance of basic cellular function, and it is found in nearly all human cells. The gene expression pattern was defined from 3 directions. Firstly, the expression breadth, which reflects gene expression on or off status. Secondly, the low or high or constant or variable expression genes based on gene expression level and variation. And thirdly, the regulation of gene expression at the gene structure level. The cancer-associated HK genes, a HK gene is specific in cancer group, while not in normal group, are expressed higher and more variable in cancer conditions than in normal conditions. Cancer-associated HK genes prefer to AT-rich genes, and they are enriched in cell cycle regulations related to function and constitute some cancer signatures. The method that was used in the experiment was RNA sequencing and using the transcriptome assembly. RNA sequencing is when snapshots are taken of RNA presence and quantity from a genome at any given moment. The experiment used 12 normal samples and 9 cancer samples to explore the general mechanisms of cancer gene expression regulation from RNA sequencing transcriptome data.
RNA sequence samples under normal and cancer conditions are selected to identify the HK genes, housekeeping genes. There are 2 issues to be considered: the amount and the saturation of the samples. Less unsaturated samples are selected as they have a lower abundance of genes, affecting the results and making the process slower. In total, 37 different cell transcriptomics are selected. Transcriptomics refer to a set of all the RNA molecules, for example, tRNA, mRNA, and so on. Of those 37, 22 were normal and 15 cancer samples selected. The normal samples include brain, spinal cortex, kidney, liver, heart, including a few others. The cancer samples include Hep G2, GM12878, lymphoma, BT474, to name a few. The samples were further narrowed down using the 5-step process above. This leads to the genes that can be used in the sample. The table shows functional enrichment analysis of normal and cancer housekeeping genes. To identify normal and cancer housekeeping gene functions, the research group compared cell gene function enrichment and signal pathways. Housekeeping genes are enriched in molecular function and biological processes. Cancer housekeeping genes participate in a cell cycle, DNA replication, repair, apoptosis pathway, along with a few other pathways to reply to tumor occurrence. Normal housekeeping genes tend to join in basic pathways. Figure A on the slide shows that cancer cell genes appear to moderate variable expressed genes. The research group investigated the ways in which gene expression variation statuses are regulated to cope with the emergence of a tumor. The group compared 6,237 shared housekeeping genes to illustrate their adjustment. The results show that more than 1/2 of shared housekeeping genes expression variation status changes between normal and cancer conditions. As shown in B on the slide, almost 2/3 of constant shared housekeeping genes under normal condition change to moderate variable status under cancer condition. 1/3 of moderate variable shared housekeeping genes under normal condition become constant shared housekeeping genes whilst in cancer condition. About 1/2 of variable shared housekeeping genes under normal condition change their expression variation status to moderate variable in cancer condition.
The The main conclusion from our research-based article would assume that the pattern of gene expression is likely more related to physiological condition instead of any spatial distance of tissue. In RNA sequencing, there was 847 cancer HK genes and 2,594 normal HK genes. So this means cells had to turn on fewer genes in cancer by upregulating them. This variable expression regulation caused diverse cancer cell types to complete regulation with a small set of genes. An explanation for upregulating is that the normal HK genes are GC rich, meaning the pairs are bound by 3 H bonds. This means they're harder to break than the AT rich cancer HK genes, bound only by 2 H bonds. At the same time, DNA needs less energy to transcribe, thus concluding that higher gene expression can be discarded from cancer-associated HK genes. You may be thinking, what was meant by patterns of gene expression being related more to physiological condition? Well, a cell type turns on lesser genes in cancer because upregulation and can complete gene expression in a smaller gene set. It just happens to be that cell types in the cancer condition did this more than those in the normal condition. A limitation of our study is that RNA sequencing gives reads from the end of a random sample of fragments. So, additional data is needed, otherwise you only get information about relative abundances. The additional data which would give us an absolute measurement includes levels of spike-in transcripts. These spike-ins are RNA transcripts used to calibrate measurements in a DNA microarray experiment. Finally, we think research like this will be of benefit to humans in understanding, in particular to our chosen article, how cancer cell types' pattern of gene expression can be differentiated from patterns of other cell types.